This is the first in a series in which we'll be taking a look at Chapter 26 of the Merriam Human Anatomy and Physiology text. We'll be talking about fluid, electrolyte, and pH balance. And really, this is an extension of Chapter 25, renal physiology. We're just going to be taking a look at the overall effects of some of the kidney functions and thinking more about the different compartments, the fluid compartments of the body. So let's take a look at the fluid compartments. Your body is composed of a lot of water. So 60% of the male body is made up of water, maybe 50% of the female body, because there's a little bit more adipose tissue, which is very low in water. We can think of the body, the fluid volumes in the body, as divided into two compartments. The intracellular compartment, so the contents of all the 37 or so trillion cells that make up your body, and the extracellular fluid, we've mentioned that a lot, the interstitial fluid, which is a catch-all term that means all the water that's surrounding, bathing all the cells in your body, and the blood plasma. There's a few other small fluid compartments in your body as well. So we're talking about a total of around 40 liters of water. Here's a little graphic that kind of gives you a visual about the dimensions, the relative size of the different fluid compartments. So the intracellular fluid, again, has about 2/3 of the water, 25 liters of water. Pretty lot of water in all those tiny little cells. And the interstitial fluid has about 15 liters of water that's bathing all those cells. And that compartment is pretty well mixed with the blood plasma, around 3 liters of volume there, since there's around 5 liters of blood with a hematocrit of around 40-something%. Solute and solvent. We talk about solutes a lot, things dissolved in water. Water is the solvent of your body. Organic chemists know there's a whole bunch of different liquids in which things can be dissolved, all kinds of compounds like xylenes and so forth, but we're just talking about water as the solvent here, and there's stuff that dissolves in water. Two categories, two sort of classes of chemicals in your body dissolved in that water: non-electrolytes and electrolytes. It's easier to start talking about electrolytes, I think. They're the ions, the electrically charged species, and most electrolytes are tiny little single atom objects, the smallest imaginable particles that have either an added electron or a missing electron, so they have a negative or positive charge, and some of them are acids and bases. Anyway, they're very, very small, relatively speaking, and very, very highly concentrated, whereas non-electrolytes tend to be larger organic molecules, big bulky things that don't have electric charge, but they're nevertheless dissolved in water, so they make up the bulk of the solutes, but they're not as numerous because the electrolytes are so tiny but so concentrated. Therefore, the electrolytes have more osmotic power because osmosis depends on the relative numbers of particles on either side of a semi-permeable membrane. And so if you have a lot of something, a large number of particles, such as electrolytes, they have a lot of osmotic power. Having a large bulk of a smaller number of solute molecules really doesn't have as much osmotic power. So your plasma is around 150 millimolar sodium chloride, maybe a little bit less than that. The plasma glucose, for example, is only 5.5 millimolar, because we're talking about big molecules, relatively speaking, but not quite so many of them. Also, electrolytes tend to dissociate, and that's not all that important for what we're talking about, but just so you know, if you took a sodium chloride crystal, it's got a lot of sodium chloride in there, all hardened up and dried up, and if you drop it in water, it dissolves, and as it does so, the sodium ions and chloride ions separate from each other. They dissociate, and for every sodium chloride, you have two species, two solute objects, a sodium ion and a chloride ion. So there's twice as many as there would otherwise be. So sodium chloride is 150 millimolar in the plasma, but there are 300 milliosmoles per liter of particles. So molarity is a measure of the number of particles of solute per unit volume. Osmolarity is the sum of all those particles, because it's a measure of what the ability of that solute is to draw water into the compartment by osmosis. So 300 milliosmoles per liter sodium chloride approximately, actually closer to 280. The total is around 300 milliosmoles per liter in the plasma. One last thing that your authors have brought to our attention is that electrolyte concentrations are often expressed in not necessarily millimolar, but milliequivalents per liter. It's a similar term. It just means that we're going to count up the number of charges, total number of charges, not just the number of objects, the number of particles. So for example, calcium ions have 2 electric charges per ion, or calcium plus 2. So when we think of the number of equivalents, it'll be twice as high as the number of particles. So milliequivalence just takes into account the total number of charges in a particular solution. Here at the top of this slide is a little description of differences between the extracellular fluid and intracellular fluid in terms of electrolytes, and the extracellular fluid has a lot of sodium chloride. As we were saying, it's around 300 milliosmoles per liter, just about. The intracellular fluid is different. There's not much sodium inside the cells. There's a lot of potassium. We've known that for a long time because we discovered that when we talked about resting membrane potentials. Cells have a lot of potassium inside, but not much potassium outside in the extracellular fluid. Extracellular fluid has a lot of sodium, but not much inside the cells. That's because the sodium-potassium pumps are working all day, every day, to create concentration gradients or differences. So that's why there's such a difference between sodium inside and outside of cells and potassium inside and outside of cells. Well, it turns out that there are always counter ions. When you have a large number of positive charges in a fluid compartment, there will also be a large number of negatively charged species of some sort. And again, in the extracellular fluid, chloride is the main counter ion, negative ion, or anion sometimes called. Positive ions are often called cations. Inside the cells, as we said, the potassium is the most prevalent cation, and hydrogen phosphate is the more prevalent anion. There are some negatively charged proteins inside of cells, quite a bit, as a matter of fact, but not so much in the way of chloride ions. So that's kind of the electrolyte composition of the cells. Plasma, the extracellular fluid, is of course 2 compartments. It's the plasma versus the interstitial fluid. They are the same in terms of electrolyte concentration, but there's a difference in protein. There's more protein concentration in the plasma than in the interstitial fluid. We know that because we talked about oncotic pressure. We said the plasma has a lot of protein and osmotic power or pressure, and the interstitium has less, and that's what partly holds water inside the leaky blood capillaries. Here's a graphical representation of the electrolyte differences between the compartments, and it's kind of great because it kind of gives a staggering view of these differences. Let's look at sodium. Sodium in the extracellular fluid, the blue and the red bars, those represent the interstitial fluid and the blood plasma together. Here's the amount of sodium out there. How about sodium inside the cells, intracellular fluid? Dramatically less. How about potassium? Even more radical. The amount of potassium in the blood plasma and interstitial fluid here in red and blue, very, very low outside the cells, and lots of potassium inside the cells, the intracellular fluid. How about chloride ion? Lots of chloride ion in the extracellular fluid, not much inside of the cells. A lot more hydrogen phosphate being the anion or counter ion inside the cells in the intracellular compartment. I guess that's all we need to say about electrolytes for the moment. The osmotic and hydrostatic pressures determine the mixing or movement of fluid and solutes between the compartments. So water moves from the plasma into the interstitial fluid by filtration. We said that there's a lot of hydrostatic pressure inside the capillaries, and they're somewhat leaky, and water and small solutes move out of the capillaries across the capillary walls and that process is called filtration. It's bulk flow. It's not diffusion. It's water pushing its way through passageways, poorly defined passageways in the walls of the capillaries. And then we said that some water moves by filtration back in at the venous end of the capillary, and there's a net loss of fluid, and that fluid is yet picked up by the lymphatic system and returned to the venous circulation. So there's this mixing going on between the plasma and the interstitial fluid in terms of nutrients and electrolytes, and a little bit less so the proteins. The proteins tend to stay in the capillaries. Water moves freely between the extracellular fluid and intracellular fluid by osmosis. So once that water filters out of the plasma into the interstitium, then water can freely cross the cell membrane into most cells. And so there's always an identity between the osmolarity of the intracellular fluid and extracellular fluid. They're always the same osmotic concentration. That's because water will always shift to make them equal. For example, if we increase the osmolality or osmolarity of the extracellular fluid, let's say we start taking away some water, you start sweating and getting dehydrated. Now we have a hypertonic extracellular fluid. Water will simply leave the cells and the cells will shrink because water moves from higher water concentration inside the cells to lower water concentration in the interstitial fluid. If we lower the osmolality of the extracellular fluid, if you drink some glasses of water, all that water goes right from the GI tract into the interstitial fluid, into the blood, and then into the interstitial fluid, then there's a hypotonic solution bathing all the cells, and water will just move by osmosis into the cells and they will swell. And that's what happens every day as you are avoiding water or unable to get some water into your system for a period of time. All your cells slowly shrink as water is lost from the extracellular compartment by evaporation, by urine formation, and so forth. And then when you drink water back, you add that water to the plasma and to the extracellular fluid, and it becomes less concentrated, and then the cells swell back up. So this is a constant thing happening between those compartments. Here's a graphical representation of those same events. There's always water and small solutes leaving the blood plasma by filtration into the interstitium, and then the lymphatic system captures the small remnant of fluid, about a couple of mils per minute throughout your entire body, and returns it to the blood. This arrow makes it look like there's a large amount. It's not the amount, it's just trying to find space to show you the direction of movements. The water moves by osmosis freely between the interstitial fluid and the intracellular fluid, the extracellular versus intracellular fluid, so that if we change the concentration of this ECF fluid out here, water will simply shift between these compartments and they will always have the same osmolality. There's also a little arrow and description of other things that are happening in terms of movement of species between the compartments. Blood gases can diffuse freely across all compartment boundaries. They are not restricted in any way, and so oxygen can diffuse from the lungs into the blood, and from the blood into the interstitial fluid into the cells. CO2 produced by metabolism can diffuse into the interstitial fluid into the blood, and then will be blown off or breathed out in the lungs and so forth. So all kinds of interactions, interchanges of solutes are represented here in this diagram. So, water balance. The amount of water coming in must always equal the amount of water going out, because our bodies stay constant in terms of water volume and solute concentrations within limits. Obviously, we saw in the very first slide, as you age, the total amount of water in your body tends to go down. But anyway, for the most part, we keep those things very, very constant. I started getting a little German in there, very, very constant, maybe Indian. Water intake is mainly in ingested foods and beverages. There's a little bit of water produced by metabolism, which is kind of cool. Brand new water molecules are formed in your body. At the end of the electron transport system, we take oxygen, hydrogen ions, and the electrons that come out, the used electrons that come out of that electron transport system, and mash them together and produce water molecules. So some water is produced that way, but mainly you take in water by food and beverages. Water output is largely in the form of urine. You can never stop making urine, as we discovered in our urinary system lab. You don't feel like you have to go, but still there's some urine being produced all the time in your body. But it also turns out that there's always water being lost elsewhere from your body. The insensible water loss refers to evaporation across your skin. Your skin does a fantastic job of preventing water loss from your body, the epidermis specifically, but a little bit of water is constantly evaporating from the surface of your skin, and every time you expire air, a little bit of water is lost in that expired air. Those are called insensible water losses. If you sweat, that's an additional way that water will be lost from the body, sometimes very, very quickly if it's hot and you're working out really hard. And also there's some water in the feces, not much. If there's a lot of water in the feces, that's diarrhea, and that's an imbalance. There shouldn't be much water in the feces. So here's again, a graphic. We can see the intake of water is largely through beverages, a little bit of water produced by metabolism, some water in the food. It's obviously not solid dry food, but it's got some level of hydration. And urine is the largest volume of water being produced in terms of loss of water from the body. Also insensible water loss, evaporation, and some sweat. Perhaps this, of course, is highly variable depending on your geographic location, the weather, your particular behaviors, and so forth, and then some water loss in the feces. But these two quantities will be equal over the long haul. So what is the regulation of the fluid volume in those compartments? Well, if you have rising osmolarity or osmolality, in other words, the concentration of your extracellular fluid compartment is going up, and at the same time, mean arterial pressure will probably be going down, because if blood volume is going down, mean arterial pressure is going down. Those are the conditions that we call dehydration. And we're going to discover dehydration as a homeostatic imbalance in just a few minutes, but dehydration, the direction of things that we call dehydration are occurring all the time in your body when you're not actually actively drinking water. So it could be a result of sweating. Or you could have a pathological condition, losing a lot of water by vomiting or diarrhea. You could lose a lot of water by blood loss. In that case, you're not actually changing the osmolarity, but you may very well be dropping the mean arterial pressure as the volume of blood goes down. That's actually called, I can't think of the word I want, so let me just come back to that. Anyway, that's dehydration. So what's happening? When the hypothalamus osmoreceptors observe this increase in osmolarity, and the baroreceptors signal to the hypothalamus about the drop in blood pressure, the hypothalamus activates the thirst response and we take in water. Also, the hypothalamus under those conditions releases ADH by way of the posterior pituitary gland. It's really just an extension of the hypothalamus, as you know. And ADH causes an increase in water reabsorption, pure water reabsorption from the collecting ducts in the medulla of the kidney. So ADH makes those collecting ducts permeable, and water is reabsorbed by osmosis because of that salty medullary interstitium. So we pee out a smaller volume of more concentrated urine and retain water and drink water. Also, ADH causes vasoconstriction, which will prop up that mean arterial pressure for us. If the plasma osmolarity is decreased as we drink water and we add some volume, some pure water volume, and it goes into the plasma, dilutes all those electrolytes and so forth, and the blood pressure will of course rise as the blood volume goes up. That inhibits ADH. And so we have less ADH and we won't reabsorb quite as much water in the collecting ducts. Interestingly, adding water into the oral cavity and then into the stomach and intestines, just stretching out those organs, inhibits the thirst response. Long before there's absorption of water into the blood and changing the osmolarity of the blood and the blood pressure, there's actually inhibition of the thirst response to some degree right away as you add water. It's kind of like a feed-forward system, it's called, so that we automatically sort of start laying off the water before we get too much on board. It takes quite a while for that water to be absorbed into the blood, so we don't want to wait until that happens to signal you to stop drinking the 12th and 13th glass of water. So inhibition begins almost immediately when you start wetting your mouth with water, interestingly. There is a disease state in terms of regulation of volumes by the way of ADH. If there's an absence of ADH, that's called diabetes insipidus. We're going to be producing a very large volume of dilute urine in much the same way that people with unmanaged diabetes mellitus do, but in this case there's no sugar in the urine, so it's called diabetes insipidus. We have a high urine flow rate, constant thirst, because we are going to be dehydrated. We can't keep enough water on board, and hypotension, can't keep enough blood volume, and then we're going to be constantly drinking a lot and so forth. So here's a little graphic that sort of lays out the things we've just been talking about in terms of the thirst response. What controls the thirst response? So if the osmolality of the plasma goes up, the concentration of solutes in the plasma goes up, and the blood volume and blood pressure go down, those things signal to the hypothalamus to activate the thirst response. Osmoreceptors in the hypothalamus, that just means as cells in the hypothalamus shrink, they signal to activate the thirst response, and so we take in some water. As water enters into the throat, the mouth, in the stomach and intestines, this little dotted line means inhibition. So it's going to feed back and inhibit right away, start inhibiting that thirst response. And as the osmolality of the plasma goes back to normal, it's going to finally obliterate that thirst response mechanism. Interestingly, when the blood pressure goes down, we activate the renin-angiotensin system. Angiotensin II, produced by that system that's designed to restore the blood pressure, also triggers the thirst response in the hypothalamus to make sure that we get the necessary volume back into the vascular system so we have enough to work with in terms of venous return and cardiac output and so forth to maintain the blood pressure. And here's another graphic showing they separated out the ADH response and the thirst responses. So again, when the osmolarity of plasma goes up and the blood pressure goes down, we signal to the hypothalamus. Here's the baroreceptors signaling to the hypothalamus. Here's the osmoreceptors in the hypothalamus. I don't know why they felt they had to separate those into two identical drawings, but don't worry about it. Same hypothalamus, and those two things combined to result in an antidiuretic hormone release, ADH release, which causes the kidneys to retain more water, reabsorb water in the collecting ducts, and we restore the fluid volume in the blood volume, and we reduce the urine volume, and we also restore the osmolality, reduce the osmolarity of plasma. Okay, so disorders of water balance. Let's quickly just look at a couple of disorders of water balance to end this first session. Dehydration. Now, I don't want you to think that the factors, the changes in conditions that we call dehydration are only taking place in the most rare of circumstances. They're normal physiological changes that help your body manage the blood volume by negative feedback systems and so forth, and osmolarity all the time. But dehydration often is used to describe a sort of advanced condition in which things have been let go too far. You haven't gotten to the water to drink water and so forth. So if you have dehydration or a clinical imbalance of dehydration, you've lost a lot of volume out of the extracellular fluid by any one of these mechanisms or multiple mechanisms. And the blood pressure is going to be very low. We've lost a lot of volume. The blood pressure is down, right? Venous return is down. Therefore, stroke volume is down. Cardiac output is down. And mean arterial pressure is down. The osmolality of the fluid extracellular compartment is high. It's going to be very concentrated because we've lost a lot of water, perhaps by, again, sweating, not taking in water, insensible loss of water, burns, water is escaping from the surface of the body, all kinds of things that may be happening to lead to a lot of loss of volume. So the symptoms we may discover are, of course, low blood pressure, but also cottony, quote-unquote, oral mucosa, kind of puffy oral mucosa, extreme thirst, dry, flushed skin, and oliguria. In other words, a very, very low urine production, abnormally low urine production, right? Because now we're massively trying to retain as much water as possible in the bottle, very high levels of ADH, vasoconstriction, and so forth. Eventually, this condition, if it's allowed to persist or allowed to degrade, will lead to confusion, and maybe even hypovolemic shock. We said when the blood pressure gets so low and the cardiac output therefore is so low that it can't meet the needs of the body tissues for oxygen, we could have hypovolemic shock, and that could be obviously a very, very dire situation indeed. But again, I just put this note down here to remind you that dropping blood pressure and rising osmolarity of plasma is what happens all the time and activates the mechanisms, the homeostatic mechanisms of thirst and ADH release to help get those conditions back to normal. keep them in their normal range. So here's a little graphic to talk about dehydration. So first thing the first step is loss of water from the extracellular fluid compartment. Here's somebody out in Death Valley running along outside there. She's probably running an Ironman out there in the hot sun because people do that kind of thing. I don't really know why, but they are drawn to it like moths to flame. But I love it out in the Southwest too, so I understand wanting to live out there. It's so beautiful. Okay, so she's out there running, she's sweating like crazy, and so she's going to lose water from the extracellular fluid compartment, and the cells are going to shrink. The cells are going to shrink as water is going to move from this now highly concentrated extracellular fluid compartment. The water is going to move from within the cells by osmosis out into here because of the rising osmolarity. How about hypotonic hydration? If we overhydrate the body, add too much water relative to the amount of solutes that are present, the electrolytes that are present, we'll have hypotonic hydration. Let's say you drink a very large number of glasses of water. Now you'll have hypotonic hydration. There was a period of time when, a short period of time when, especially when there's pressure on colleges and on fraternities and sororities at various colleges to reduce the amount of binge drinking and using alcohol as a hazing tool. And so somebody came up with a genius idea. What if we punish our pledges for hazing by making them drink vast quantities of water? It's not any kind. They won't get any kind of impairment of their body or their judgment and their coordination and so forth. It'll be perfectly safe. Who could complain about that? Well, the people became hypotonically hydrated. It's called water intoxication. And another time this can happen is with renal insufficiency. If your kidneys aren't working properly and you can't manage to reabsorb the right amount of electrolytes, you're going to have possibly hypotonic hydration. So anyway, the osmolarity now, or osmolality of the extracellular fluid in this case is going down, blood pressure is going up, and so water moves into the tissues and the cells swell. And they swell and potentially continue to swell. This can cause some severe metabolic disturbances, including nausea and vomiting, can cause muscular cramping. If someone has severe hypotonic hydration, possibly because of, again, renal problems or this condition we talked about, a manual taking on of water, you can add some hypertonic saline, ingest some hypertonic saline or infuse some hypertonic saline, and that will help restore the electrolyte balance in the body. But one interesting thing, in extreme cases, the cranium is a bony box that has a limited or defined volume. And if the trillions of cells that are in your cranium expand because of hypotonic hydration, the pressure inside the cranium begins to rise. And the only way blood can be driven up into the brain is by raising the arterial blood pressure higher than the intracranial pressure, and that's exactly what happens. The blood pressure begins to skyrocket, and every time the blood pressure goes up, there's even more edema that's taking place in the cerebrum because of high hydrostatic pressure in the capillaries and so forth. There's this runaway condition of higher and higher intracranial pressure and cerebral edema and loss of circulation, blood circulation into the brain, which can eventually lead to convulsions and death. And actually some college students actually died from hypotonic hydration in fraternity and sorority pledging. I think mainly fraternity pledging. I think women are a little more sensible in many ways. So anyway, this very rapidly halted the popularity of this pledging strategy. Anyway, if this happens, you can use, interestingly, hypertonic mannitol. If you infuse hypertonic mannitol, it's a sugar that can't escape by way of capillary filtration into the interstitial fluid in the brain and the cerebrospinal fluid. So it's retained in the blood, and because it's hypertonic, it will draw water by osmosis into the vascular system from the intracranial volume, and you can reduce that intracranial pressure. Kind of cool. So here's a little graphic again showing hypotonic hydration. So if we take in too much water, or the kidneys are at fault for not being able to retain enough solutes relative to the amount of water as they function or fail to function, we have a dilution of the extracellular fluid, a hypotonic condition, and so water moves from high water pressure, high water concentration to lower water concentration in the cells, and they swell. Finally, edema is the accumulation of tissue water. In the interstitial fluid in the tissues, the water that's present is a constant fixed amount, and it's constantly entering through from the plasma and being carried back into the blood by the lymphatic system. But if the amount of fluid arriving in the interstitial fluid in the tissues exceeds the rate that the lymphatic system can scavenge it up, there will be edema, there will be swelling in the tissues. So, what can cause edema? One is increased, there's two forces that control capillary filtration. That's the source of the water, the blood. If we increase the hydrostatic pressure, which is the outward force pushing fluid out of the capillaries, we'll have potentially edema if if the fluid accumulation exceeds the rate that the lymphatic system can scavenge it up. Hypertension, you have high blood pressure. High arterial pressure means there's going to be higher capillary blood pressure, higher hydrostatic pressure, a greater filtration rate. So for example, left heart failure causes pulmonary edema. Congestive heart failure is all about pulmonary edema happening because of the backup of blood into the lungs, increased hydrostatic pressure in the capillaries of the lungs, and fluid accumulation. If you have right heart failure, if the right heart experiences overwork because of, say, emphysema, years and years of emphysema, high resistance to flow in the pulmonary vasculature, or a valve defect, possibly, that could cause overworking of the right heart. Those things can cause right heart failure. Then you'll have blood backing up into the systemic circulation, the systemic capillaries, and you'll have systemic edema. Another thing that can cause edema is excess blood volume. That will just push more, create more hydrostatic pressure, and that'll cause more fluid to leak out. The other force that governs capillary filtration is oncotic pressure, the protein concentration in the capillaries that holds the water essentially in. But if you raise that oncotic pressure, let me turn around and say that again. If you lower that oncotic pressure, so it's not holding the water in the capillaries, that's going to increase the rate at which water escapes, and we're going to have possibly edema formation. So when would the protein concentration in the blood go down, the oncotic pressure go down? It'll go down when you don't have a source of plasma proteins. If there's sufficient inflammation in the liver or liver disease that the liver can't produce enough plasma proteins, then there's going to be a decrease in oncotic pressure, and there's going to be an increase in capillary filtration. So people with liver disease tend to have systemic edema. Malnutrition, if your protein in your diet is so low that you can't produce the plasma proteins, there's not enough amino acids, not enough raw material to make those plasma proteins, then we're going to have a lower oncotic pressure and higher filtration rate and edema. And finally, glomerulonephritis. One of your homework questions asks about proteinuria. There shouldn't be protein in your urine. But if you have glomerulonephritis, it's very likely that there's going to be protein in the urine, and your liver will not be able to keep up with the rate at which protein is being lost in the kidneys at the failing filtration membrane, and you're going to have reduced oncotic pressure. So people with this type of kidney disease tend to have, again, edema and difficulty in keeping the volume in the blood, keeping the blood volume intact. Inflammation. Histamine is a vasodilator, and we know that vasodilation increases capillary hydrostatic pressure. We're opening up those arterioles and letting more of the mean arterial pressure into those capillaries. Hydrostatic pressure goes up and filtration rate goes up. Interestingly, histamine also increases the permeability of the capillaries and venules, and so the rate of leakiness is greater at a given hydrostatic pressure, and so there's just a lot of water that's escaping from the vasculature in times of inflammation. Finally, sometimes we remove lymphatic vessels. We remove lymph nodes. Have you ever heard of that? People sometimes, when they have different types of cancers, breast cancer, for example, may metastasize, and cancer cells take up residence in the lymph nodes and lymphatic system of the axillary region in the thoracic region, and those lymph nodes will probably be removed surgically at the time of remediation of that cancer. And so then there may be a lot of problems with edema after that because of a loss of the normal lymphatic mechanism of getting water back into the blood from leaking out of blood capillaries. And that doesn't happen just with breast cancer. There could be various types of lymphomas that involve removing some parts of the lymphatic system, other types of cancers that some of part of the lymphatic system may be removed during radical surgeries, or specifically to remove infected or cancerous lymph nodes. So anyway, blocked lymphatic vessels or absent lymphatic vessels can cause edema. All right, when we come back, we'll talk about, for our second session, we'll talk about electrolyte imbalances and then go on from there as we proceed through chapter 26. So see you next time.